Welcome to this lecture on glucose metabolism and the role of aldolase. After this lecture, you will be able to describe the general mechanism of gluconeogenesis and glycolysis, describe the function of aldolase in both gluconeogenesis and glycolysis, describe the function of the aldolase B isoform, and you'll be able to describe the regulatory action of transcription factors in aldolase B expression. Firstly, we will review the process of glycolysis, which occurs in the cytoplasm of the cell. Glycolysis is the process of glucose breakdown for the eventual extraction of ATP energy. It is the first part of cellular respiration. And the products of glycolysis includes pyruvate, ATP, and the energy carrier NADH. It is important to note that glycolysis is a multi-step reaction that is composed of several enzymes that catalyze each step. In the first step, hexokinase catalyzes the conversion of glucose into glucose 6-phosphate. In the second step, phosphoglucose isomerase catalyzes the conversion of glucose 6-phosphate into fructose 6-phosphate. In the third step, phosphofructokinase catalyzes the conversion of fructose 6-phosphate into fructose 1,6-biphosphate. In the fourth step, aldolase catalyzes the conversion of fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, or DHAP and G3P for short. And in the fifth step, which is not shown here, triose phosphate isomerase catalyzes the conversion of DHAP into G3P. In the sixth step of glycolysis, G3P dehydrogenase catalyzes the conversion of G3P into 1,3-biphosphoglycerate. And in the seventh step, phosphoglycerate kinase catalyzes the conversion of 1,3-biphosphoglycerate into 3-phosphoglycerate. And in the eighth step, phosphoglyceromutase catalyzes the conversion of 3-phosphoglycerate into 2-phosphoglycerate. And in the ninth step, enolase catalyzes the conversion of 2-phosphoglycerate into phosphoenol pyruvate. And in the 10th and final step, pyruvate kinase catalyzes the conversion of phosphoenol pyruvate into pyruvate. So it's important to note that the majority of these reactions are actually isomerizations or phosphorylation type reactions. Certain steps of glycolysis are highly regulated with tightly regulated enzymes. These enzymes include hexokinase, phosphofructokinase, and pyruvate kinase. This is a more detailed representation of glycolysis, and as you can see, the products are being formed in different steps of glycolysis. So our products are going to be, again, ATP, and NADH, and pyruvate. On the other hand, the process of gluconeogenesis can be considered the reverse of glycolysis. The purpose of gluconeogenesis is to synthesize glucose, and to do this we need other non-carbohydrate precursors or products of glycolysis, which in this case is pyruvate being used to synthesize glucose. Other examples of non-carbohydrate precursors can include amino acids, fatty acids, and glycerol. This diagram shows you glycolysis and gluconeogenesis side by side to show you that they share a similar pathway, but it is in reverse. So glycolysis is going to be the breakdown of glucose for the formation of 2-pyruvates, and gluconeogenesis is going to be utilizing pyruvate or other non-carbohydrate precursors for the synthesis of glucose. So it is going in the opposite direction. So we are going to talk about aldolase now, which is the enzyme that is going to catalyze step 4 of glycolysis. So aldolase is responsible for catalyzing the catabolism of fructose 1,6-biphosphate into dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. Aldolase is also responsible for the reversed anabolic reaction as well. This is a general mechanism of the reaction that aldolase helps to catalyze. And this mechanism or reaction can be considered an aldol cleavage. And it's important to note the catalytic residues that are located in the active site of the aldolase enzyme. And these catalytic residues are important because they facilitate the catabolism of fructose 1,6-biphosphate into DHAP and G3P. One of these residues is lysine, which is very important in the formation of something called our shift base intermediate. And our shift base intermediate is very important in breaking carbon-carbon bonds and for the eventual catabolism of fructose 1,6-biphosphate. So the shift base intermediates here on our mechanism right here are going to be located right here. Here's one of them. And our shift base intermediate is going to reform here again. And they really facilitate the reaction because they increase the reactivity. And next, we are going to talk about the different aldolase isoforms. So in vertebrates, there are 3 different isoforms of aldolase. The first type is called aldolase A, found in skeletal muscles, erythrocytes, and other cells. So here's a model of aldolase A. The second isoform is aldolase B, which is found in the liver, kidneys, or intestines. Aldolase B right here. Aldolase C is found predominantly in the brain. So the model is here. So all 3 isoforms are part of the class 1 fructose biphosphate aldolase gene family. So to talk further about the different isoforms, aldolase A is expressed by the ALDO-A gene. And the ALDO-A gene is located on chromosome 16 on the P arm at position 11.2. The active site of aldolase A includes 2 notable residues, lysine in position 230, which helps in the formation of our shift base intermediate, and glutamic acid at position 188, which acts as a proton acceptor, or it mediates the transfer of protons. Mutations in ALDO-A leads to certain diseases, and an example is called glycogen storage disease 12. And also, mutations in ALDO-A is linked with several cancer progressions. On the other hand, aldolase C is expressed by the ALDOC gene. ALDOC is located on chromosome 17 on the Q arm at position 11.2. The active site also includes the conserved amino acids seen in aldolase A and actually seen in all of the isoforms. Last but not least, we will talk about aldolase B, which is expressed by the gene ALDOB. ALDOB is found on chromosome 9, and it's located on the Q arm at position 31.1. Its active site also includes the notable amino acids, which are going to facilitate the eventual catabolism of fructose 1,6-biphosphate. Mutations in Aldolase are going to lead to fructose intolerance and eventual liver disease, and this is caused by an accumulation of excess fructose 1-phosphate in the liver, which is going to lead to hepatic diseases. So like all the other isoforms, aldolase B is going to help catalyze the reaction in step 4 of glycolysis. So it's going to catabolize fructose 1,6-biphosphate into DHAP and G3P. But aldol or aldolase B is also important in the process of fructolysis. So aldolase B is going to help catabolize fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate in fructolysis, which is the breakdown of fructose instead of glucose in glycolysis. So aldolase B, right here in the diagram, is going to catalyze the catabolism of fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate. And both are going to be converted into glyceraldehyde 3-phosphate with the help of other enzymes. And glyceraldehyde 3-phosphate can be used to create fatty acids, eventually create ATP or lactate. Or glyceraldehyde 3-phosphate can be converted into fructose 1,6-biphosphate, eventually being synthesized into glucose in our gluconeogenesis pathway. Last but not least, we will talk about the different transcription factors that regulate the expression of the Aldolase gene. So the first transcription factor is called HNF1, which is a very strong transactivator of the Aldolase gene promoter. And on the diagram, as you can see, it's right there. HNF3, on the other hand, is interesting in that it is a very strong anti-activator of the aldolase B promoter or Aldolase promoter. It does so by competitively antagonizing the transactivation of HNF1 and non-competitively antagonizing the transactivation of DBP, which is another transcription factor. So HNF3 is located there on the diagram. CEPB is another transcription factor, which is not as strong as HNF1. It is a weak transactivator. And lastly, DBP is a strong transactivator of the Aldolase gene promoter. So after this lecture, you learned that glycolysis is essential for generating ATP from the breakdown of glucose. You learned that gluconeogenesis is the process of converting non-carbohydrate precursors to glucose. You learned that aldolase is a key enzyme that catalyzes the interconvertible catabolism of fructose 1,6-biphosphate, or fructose 1-phosphate, in glycolysis or fructolysis. Also, you learned that aldolase B expression can be regulated by several transcription factors acting on the Aldolase gene.